life in television, I learned early on that news, generally speaking, is bad news. Good news is public relations. It's advertising. And so the question arises, is there any good news out there? And generally speaking, in our society, when we read of good news, it involves technological or scientific breakthroughs. So CIFAR, the Canadian Institute for Advanced Research, is the vehicle by which Canada supports leading-edge research. Zillions of dollars are involved, and the range of subjects is breathtaking. And as an antidote to usual complaints about lack of transparency or misspent money, the people at CIFAR, several of whom are in the audience today, are eager to let Canadians know what their grantees are up to, and they have supported us not only with a little bit of sponsorship, but also by way of introducing and inducing some of these exceptional researchers to come up here and let you know what they're doing. So Steve Scherer is a leader in the field of genomics as it can be applied to personalized medical treatment. Steve says that your sequence will, in the not-too-distant future, be available to you on your BlackBerry or on a credit card. And the question arises, if you could see that far into the future, would you want to know? Steve?
Well, thank you very much. It's a great pleasure to be here. As an academic scientist, we worry about our next publication, losing our grants, in some cases scientific fraud. But today I really worry about having to present after a Yiddish rapper. And I just don't know how to deal with that, so I will do my best. I will argue today that knowing your genome is like knowing no other information. And our species has already evolved to the point that we have the ability to sequence our own genetic blueprint. And I would argue that really having this information for the first time, we'll be able to truly know ourselves. Importantly, society craves information, and your genome is the ultimate form of information. It tells you about your past, your present, and your future. And it's a binary code of data. It's actually quite easy to handle once you have the information. I'll talk about how much data there is and how large it is, but in fact, when you have the sequence of your genome, you can carry it around on a CD or, in theory, on your BlackBerry. And importantly, we think that technology is poised to make the genome a standard medical test. In the same way you have observational interviews, MRIs, for example, you also carry around your DNA sequence to complement any other types of medical tests. And we think we can tie all of the different medical information to the DNA sequence itself. And so the Human Genome Project was an initiative put together over a 10-year period to decode the order and identity of all of the chemical bases of that information. One of the very interesting observations that came out of the Genome Project was that all of us in the world population, or if you compare the DNA sequence of any two individuals in the population, regardless of their geographical, ethnic, color, background, would be 99.5% identical. Even George Bush and Osama bin Laden, 99.5% identical. So what that means is we have a common humanity. The vast majority of our variation accumulated when we spent the most of our time in Africa. And we only migrated out of Africa some 100,000 years ago or so. So if you look at any genetic variant in any of our genomes, in 90% of cases, it will be a common variant that all of the population carries. And as we migrated across the world, we collected new genetic variations. Okay, so about 10% of the variations will be specific to different groups of people. So why do we study genetics? Well, genetics plays a role in every disease that we know about. Okay, genetics has some type of a role. In some cases, it's absolute. If you have a specific genetic mutation in a gene that's involved in cystic fibrosis, you will have cystic fibrosis. Huntington's disease is another example. There's over 6,500 genetic diseases cataloged to date, and as a community, we've identified about 2,000 of those disease-causing genes. So we've made a lot of progress now, but we still don't have them all identified. 9 out of 10 of the leading causes of global death have some type of a genetic component. I've just listed heart disease as one example, but diabetes, we heard about this morning, obesity, they all have genetic contributions. In some cases, single genes can be involved. In some cases, it's combinations of genes and environmental cues, like diet, for example, or smoking. The more and more we study genetics, the more and more we learn that, in fact, genes play a role in cognitive and behavioral conditions. And my laboratory studies autism, and I don't have a lot of time to talk about it, but we've now identified genetic lesions along chromosomes and specific genes that can account for about 10% of autism spectrum disorder. So we can use this information, of course, in diagnostic testing, and we can use it to actually try to identify therapeutics. And in some cases, for example, in the case of autism, if you have early identification, we can think about applying the information for early intervention through behavioral training or language training, for example. And I'll give you a few other examples as I go forward, but I could stand up here and give you anecdotal stories of how this information is being used clinically, but I think all of you know that genetics plays a role in disease. So the current healthcare paradigm is one like this, where you have diagnosis, monitoring, and then treatment. And we think going forward, now that we have the ability to look at DNA sequences en masse, it will look something like this, which will be the new healthcare paradigm. We have genetic information determining sequences of genes or entire genomes very early on, leading to earlier diagnosis, perhaps preventative action, either through modifying your lifestyle, modifying your diet, for example, or perhaps taking therapeutics. And ultimately, finding the disease-causing genes leads to us to develop rationalized drugs, treatments, and therapies, ultimately improving survival or quality of life. So this is a happy talk, not like the talks we heard about this morning. We think that genetics, if used properly, can be an information source that helps us guide our lives. Now, technology is poised for what I would consider to be the perfect experiment. Most of the experiments we do in the genetics laboratory are really imperfect. They have flaws in that we can't look at the complete genome sequence yet, although we're almost there. So if we try to do an association study with DNA sequence in a particular trait or disease, we always have problems with statistical noise because we can't gather all of the data. And what we do in the laboratory and in the clinical setting is for 50 years, we've been looking at 46 units of information. These are the chromosomes that have banded patterns. So chromosome 1, this is the so-called karyotype from a male. You have an X and a Y chromosome. And more recently, we've been able to look at the genome using microarrays or gene chips. And last summer, a new set of microarrays came out from a few different companies that allowed us to look at about 1 million data points across the genome in one experiment for about the same price, about $500, the same price as doing a karyotype. So we increased our resolution several orders of magnitude to scan the genome. And now we're poised at a time where we can actually generate the complete genome sequence of any individual in the room. And right now, as I'll show you a little bit later, probably would cost us about $150,000 to do this, but we think the price is going to drop rapidly. So the first DNA sequence of an identified individual, Craig Venter, a scientific entrepreneur in the United States, was published in September of last year, and our group is involved in this project. James Watson, the co-discoverer of the double helix, he has his genome sequenced, and there's a female and an Asian, and there's 1,000 other projects on the go right now. So sequencing is happening very, very quickly. There is an X Prize that is challenging the community to generate 100 human genome sequences in 10 days, which would bring it into the $1,000 sequence range. So that actually will happen over time. I mentioned currently it costs about $150,000, and I work at the Hospital for Sick Children. We're actually preparing for a day where children or parents will be offered a service of having their child's complete genome sequence before the child leaves the hospital. So how do we deal with that information? How do we use it to better the medical health of that individual? Even the most effective drugs on the market are only effective in about 60% of the population. And actually, in most cases, it's much less than that. And the reason for that, at least one reason, is because of our genetic profiles. We have different genetic sequences, produce different proteins, they interact with drugs in different ways. So if we can identify up front different groups of the population that will respond in different ways to different drugs, so those having desired response, no response, and most importantly, those that will have toxic side effects. In North America, hundreds of thousands of deaths occur every single year because of an adverse reaction to a drug that they're taking. So if we can identify this up front, we will then actually be able to modify the dose of a drug or give a different class of drugs to the individual. And that's the idea behind pharmacogenomics. So I mentioned a lot about the information of the past, the present, and the future. This is an example of an individual family where we were able to identify a disease-causing gene that leads to the development of a very severe form of epilepsy called progressive myoclonus epilepsy. So these children are born, they live 10 years completely normal lives. They just look like healthy little kids. And then at about age 10, they severely deteriorate, have grand mal seizures, they're wheelchair bound, and all pass away within 10 years. So the mother contacted us because she had a child with this form of epilepsy who was 18 years old. So she knew that this boy had epilepsy. She had an 8-year-old female little girl who hadn't reached age 10 yet, so she didn't know if the child would actually have epilepsy or not. But that's not what she wanted to know. She wanted to know if the fetus she was carrying would become an epileptic child or not. Using in vitro fertilization, we can select eggs and sperm, and then the fertilized egg, as the fertilized egg grows to 8-cell stage, we just slough off a single cell, use a technique to actually amplify up the DNA and test to see if that fertilized cluster of eggs is carrying both copies of the recessive mutation. And then by selecting one healthy chromosome, you would just need to have either one or yellow or blue chromosome. You can have a child that doesn't have this type of epilepsy. In the West, we think of the individual. We tend to think of our genome as our information, right? It belongs to us. And when you're doing genetic counseling for families, it's very non-directive. So you give them the information and the family makes the decision. But in the East, it's actually the other way around. The genome is part of the heritage of the community. The individual is part of a larger community, and typically decisions are made in the larger context. As we understand more and more of our genomes, and with all the external pressures we heard about this morning, I think we'll start to see a merging of these two philosophies. And very interestingly, I think we'll be able to really get to this question of, can we quantitate nature versus nurture? This is what we all do as scientists every day. We study this relationship of your innateness and your nurture. What is the contribution of each? And I'll just close with a story that I think will just, remind me that our genes are not everything. I was a scientific consultant on a documentary called After Darwin, and we had the opportunity to go and visit Dolly, who was the first cloned sheep. Okay, so an udder cell was taken from her mother, her biological mother, and using nuclear transfer, this sheep was generated. An experiment I was told would never actually work. So we went to this farm actually outside of Edinburgh, and I'd never been to a sheep farm. And I went to the barn, and there was all these sheep. They all looked the same to me. So which one is Dolly, and who really cares? And so I went up to the pen, and all these sheep were just wandering around. One sheep came running out and came running towards us. And in fact, it was the sheep that had been raised around cameras, recognized the camera crew coming in. So Dolly was not interested in anything other than the camera. It wasn't interested in the camera. It was interested with the food that came along with the cameras. And she started to eat the cheesies out of the bag that I had, and ate a whole bag of cheesies while we were standing there trying to take pictures of the sheep. So if you read in the press about Dolly dying because her telomere shortened, or she had this or that, it was really her diet. And I'm absolutely convinced. So it's the first cloned animal where you think genetics would play the complete role in this animal's life. The same with identical twins, for example. Yet in fact, it was environment that actually knocked her down. So I would close on that thought, just reminding you that there's complexity in every issue that we study. Thank you very much.